everyone, my name is Sarah. I make content about nursing school. Right now I'm a pre-nursing student. I am taking the ATI TEAS exam in 5 days. So I'm very nervous. Today I'm going to be going over my science review. This is to help me study and also to help you review because I learned by talking it out. So I'm going to be going over my notes. Today we're going to be going over basically everything that is in anatomy. So this is part 1. I'm going to be doing my review in 3 parts. Part 2 and 3 is going to be splitting up anatomy subjects, but part 1 is going to be everything that's not anatomy. So we're going over genetics, heredity, cell biology, cell division, chemistry, all that good stuff. So I also time-stamped all of the sections. So if you want to skip to one part of the video, you can do that. It's all in the description. And then also my notes that I took, they're going to be more in depth than what I'm going over in this video, just because I'm condensing everything to the important things. But if you want to access my notes, they're for free in my blog at slightlysour.com. I just wanted to thank Hazel and Simone for always commenting on my videos. I see you guys. I love you. Mwah. Thank you so much for commenting. I super appreciate you. So I just wanted to say that really quick before I start this video because it really does mean a lot. I read your comments and it's so nice to see people engaging with things that I put out, and I love it so much. So thank you. So let's start with cell theory. There are 3 parts to it. One is that cells are the fundamental unit of life, or fundamental unit of structure and organization. 2 is that all cells come from pre-existing cells. You can't make a cell out of nothing. It has to basically come from a parent cell. And then 3 is that, sorry, I'm remembering. 3 is that all organisms are made out of at least one cell. Most of the time more cells, in our case, we're multicellular organisms, so we are made out of many cells, but all organisms are at least made out of one cell. So now we're going to go into cell biology. What you need to know is that cells are either prokaryotic or eukaryotic cells. Prokaryotes are single cell organisms, mainly found in the bacteria and archaea domains. They don't have a nucleus. That's their defining characteristic. Also, some things to know about them is they tend to have flagella, which are little string looking hair-like structures that help with their movement. And then they have pili, which are little it looks like stubble, kind of like peach fuzz. Those are on the outside of the cell as well, and that helps with transferring DNA between other cells and just general intracellular communication. Eukaryotic cells, which is pretty much what you're tested on for the most part from what I gather, that's going to be what I talk about from here on out. I'm referring to eukaryotic cells. Those are going to be plant and animal cells. They have a nucleus. They have organelles, which we'll get into in a second. And then let me just make sure I'm not missing anything. Yeah, they have a nucleus and they have organelles. That's what you need to know. They're found in multicellular organisms. We went over that. Plant and animal cells. Cool. Okay, so now we're going to get into the organelles. And if you see me looking down, I have my laptop with my notes open. So that's why I'm looking down. So I'm going to try to list as many off from memory, and then I'm just going to fill in the gaps. So you need to know that the nucleus houses DNA. Inside of the nucleus is a nucleolus, and that is where ribosomes are made. The nucleus, in between the nuclear membrane, the outside of the nucleus, and the nucleolus, which is the innermost part of the nucleus, is nucleoplasm, and that's where DNA is. So then ribosomes make proteins, and then surrounding the nucleus is the endoplasmic reticulum, which comes in 2 forms: the rough endoplasmic reticulum, and then the smooth endoplasmic reticulum. I'm just going to say smooth ER and rough ER because it's a lot of words. The rough ER has ribosomes, so they're going to be involved in making and processing proteins. And the smooth ER makes lipids. And then we have the Golgi apparatus, which is kind of the FedEx center of the cell. It's going to be involved in processing, packaging, and delivering materials, mainly proteins like enzymes. And let me make sure I'm getting all this. Yeah, modifies and processes enzymes, which are forms of proteins, and sends them to other parts of the cell in form of vesicles. The little folds of the Golgi apparatus are made of like cisternae, I believe, and then they bud off, they kind of like pinch off to form vesicles, like that's what gets sent. So kind of like the FedEx hub of the cell. And then everyone knows mitochondria is the powerhouse of the cell. That's what generates the energy in the form of ATP. And then we have lysosomes. I remember lysosomes like Lysol, it breaks down things. So like kind of like how Lysol breaks down germs, lysosome is going to break down cellular waste. It's going to recycle materials, all that good stuff. Vacuoles are going to be mainly used for storage, like water storage. I'm pretty sure they also store like nutrition. Oh, they store water and cellular waste. Just kidding, not nutrition. LOL. Let me see what I missed. Okay, so cytoskeleton gives the cell its structure. Cytoskeleton is made out of microtubules, microfilaments, and intermediate filaments. And then the filaments themselves, not necessarily part of the cytoskeleton, but like the filaments actually also help in like movement of things around the cell, or like inside of the cell. So that's fun. Right? Yeah, movement and transferring of materials. And then we have, what did I miss? I know I missed something. Oh, cell wall is specific to plant cells. That helps with its structure. It's kind of like the nuclear membrane, kind of for animal cells. Nuclear membrane helps balance what comes in and out of the cell via selective permeability. And then kind of similar to mitochondria for animal cells is the chloroplast for plant cells, and the chloroplasts are responsible for making energy in the form of ATP for plants. So chloroplast is like very much like a mitochondria, and then the cell wall is very much like a cell membrane, also known as a plasma membrane. And I still know I'm missing something. I feel like I'm missing, what am I missing? I'm just reading through everything. Lysosome. No, I think I got everything. Those are all the notes that I took on that. And again, if I missed something, it's going to be in my notes, which I post on my blog. So we did that. Now we're going to move on to macromolecules. And what you need to know is that there are 4 of them. They're also called biomolecules, but for my sake, I just wrote macromolecules in my notes. So the 4 of them are lipids, carbohydrates, nucleic acids, and proteins. And then the main part that you need to know about these is what the difference between a monomer and a polymer is. A monomer is basically the building block of a macromolecule, and then a polymer is like a chain of monomers. So for example, for carbohydrates, the monomer of a carbohydrate is a monosaccharide. Think glucose, fructose, galactose. The polymer of a carbohydrate is going to be a disaccharide or a polysaccharide. So I'm pretty sure like sucrose is a glucose molecule and a fructose molecule. Like that makes up sucrose. Cellulose is a polysaccharide, so that is like what I'm pretty sure the plant cell wall is made out of. I think I could be wrong. Don't think you're going to be tested on that, but it's probably good to know. Lipids. So lipids they're not true polymers, but the monomer of a lipid is a fatty acid, and then polymers are going to be like glycerol molecules plus fatty acid. So it's not like a true chain of monomers, but like still good to know. Proteins, you do need to know that the monomer of a protein is an amino acid, and then a polymer is a like amino acid chain or a polypeptide, like that's what it's called. And then for nucleic acids, a nucleotide, or in some, I think the ATI manual put it as something else, but what I gathered was that a nucleotide is the monomer, and then polymer is DNA and RNA. So now I'm gonna skip over cell respiration. It was in one of my study books, but it wasn't in the other 2. So I'm going to assume that it's not important information to know, but again, if you want to know, it's in my notes on my blog. Photosynthesis, I'm also going to skip over. I don't think that's very important information, but we're going to move on to cell reproduction, which is, so there are 2 types of cellular reproduction. There is sexual reproduction and asexual reproduction. Asexual reproduction is pretty much just an exact copy of a cell. So like when a cell just like splits into 2, like one cell that like splits itself into two. And then sexual reproduction is when two parent cells come together and then they form new cells. So the sexual reproduction is going to allow for genetic variability. It's going to allow for changes basically, and then asexual reproduction is going to be everything stays exactly the same unless there's like a mutation or something, but pretty much everything stays the same. And then for asexual reproduction and then for sexual reproduction, that is where the genetic variability comes in. So asexual reproduction is called mitosis. It's broken up into 4 phases, which starts with, I should probably go over the cell cycle first. Okay, so so basically, cell cycle is G1, S, G2, and M. G1 is cell growth, so the cell is duplicating its organelles. The S phase is DNA replication. The G2 phase is getting ready for division, making more organelles, and then protein synthesis, I'm pretty sure. And then the M phase is active cell division. So the M phase, we're going over mitosis. Mitosis is broken up in 2 phases, which are prophase, metaphase, anaphase, and telophase. Following all of that is cytokinesis. So prophase is when the nuclear envelope, like we talked about, like the nucleus, the nuclear envelope is degrading and the DNA is condensing into visible chromosomes. Pretty much also the centrosomes migrate to opposite ends of the cell. Metaphase is when the chromosomes are moving to the metaphase plate, like the center of the cell, and the spindle apparatus, aka microtubules, are pushing those chromosomes to like line up. Anaphase is what happens when the chromosomes are perfectly aligned along the metaphase plate, and then the separatin pulls the chromosomes apart into sister chromatids. So like chromosomes looks like, I can't do it properly, but chromosomes like looks like this, and then now they look like this, like they're being like pulled apart. Or maybe in the diagrams I show them like this. So we're gonna say that they're pulled apart to opposite ends of the cell. And then telophase is when the nuclear envelope reforms and then the chromosomes are decondensing. And then cytokinesis is when a contractile ring forms around like the 2 cells that are now coming apart, and it like completely splits them apart, forming 2 new daughter cells. So that is mitosis. And then meiosis is the, again, sexual reproduction part. So this is when 2 different cells come together, and it has 2 divisions. So the reason why it has 2 is because in mitosis, chromosomes are being split into sister chromatids, but in meiosis there are chromosome pairs that are first split, and then the pairs need to split into chromatids. So it happens twice. Hopefully that wasn't too confusing. Because the 2 cells are coming together, then you have 2 sets of chromosomes, and then those split, and then the 2 sets of chromosomes need to also split, so it forms like 4 cells basically, whereas mitosis, it forms 2 because the cell is like splitting itself. So hopefully, I don't know how to like, I understand it, but like I'm trying to say it in a way that people can understand, so I'm sorry if like it comes off really confusing. Meiosis happens in 2 stages, meiosis 1 and meiosis 2. Pretty much it follows the same like phases as mitosis. It just has a 1 or a 2 after, depending on which stage of meiosis it is. So prophase 1 is when the nuclear envelope is degrading, and then the 2 sets of chromosomes condense. And then while this is happening, I think it's called synapsis. This might be going too in-depth for what the TEAS actually tests, but it's still good to know. So synapsis is when the chromosomes are condensing into each other, and then genetic crossover happens. And this is what allows for genetic variability. So basically the chromosomes, I'm like picturing in my head, they'll like swap information on the same spots. So then they both come out differently, basically. So there you go. And then prophase is when, again, the centrosomes, migrate to opposite ends of the cell. Metaphase is when the chromosome pairs they line up along the metaphase plate. Anaphase is when the chromosome pairs are split, so they're still like, that X-shaped thing that you're thinking of. They still look like that. So now they're splitting. And then telophase is when the chromosomes decondense, the nuclear envelope reforms, and then cytokinesis, the cells are split into two via the contractile ring. And then now we kind of are, from this point on, it's going to look a lot like mitosis. So instead of going back into the cell cycle, because we talked about the cell cycle is G1, S, G2, M, and it goes back to G1, it's going to skip that, which is also known as interphase. Interphase is G1, S, and G2. It's going to skip interphase and then go right back into prophase 2. So prophase 2 is when the chromosomes are going to, or the DNA is going to condense into chromosomes. Then you have metaphase, which is the chromosomes line up along the metaphase plate. Anaphase, separatin pulls the chromosomes apart into sister chromatids now. And then telophase is when the sister chromatids are decondensing, the nuclear envelope reforms, cytokinesis, contractile ring. I picture it like this. The contractile ring forms, splitting them now into 2 sets of 2 cells. So now you have 4 haploid cells. So for mitosis, you have a cell that splits into 2. It just splits itself. And then for meiosis, you have 2 haploid cells coming together to form a diploid cell. The diploid cell is going to split into 2 haploid cells. And then those 2 haploid cells are going to split, and then those are going to form 4 haploid cells. When this happens to form egg cells, it's called oogenesis or oogenesis. This is going to form 1 mature egg cell and 3 polar bodies. And then spermatogenesis is when it forms 4 sperm cells. I'm going to skip over DNA replication and protein synthesis. If you want to know more about that, again, it's in my notes. We're going to move right along into genetics. So basically about genetics, what you need to know is that Gregor Mendel is considered the father of genetics, based off of his experiments with pea plants that developed the modern understanding of heredity. So a gene is described as like a trait that someone inherits from a trait that something inherits from its parents. So those traits from the parents are called alleles. So gene equals allele 1 plus allele 2, 1 coming from each parent. So a genotype is the set of genes that someone carries, and a phenotype is the physical expression of that gene. Genes can be dominant or recessive. So the dominant is what is expressed, and the recessive is what is not expressed, pretty much. I could be wrong though, so someone please correct me in the comments if I'm wrong. So the recessive trait is only expressed when it's a I can't say this word, which is why I'm looking at it. Homozygous recessive genotype. So basically what that means is homozygous means that the 2 alleles are the same, and then heterozygous means that the alleles are different. So when it's homozygous dominant trait, you're going to see 2 capital letters. When it's a heterozygous trait, you're going to see 1 capital and then 1 lowercase. And then when it's a homozygous recessive, you're going to see 2 lowercase letters. So when you're looking at a Punnett square, for example, if a question asks, what is the chance that this particular trait will be expressed? If it's a recessive trait, it's when you only see those 2 lowercase letters together. And if they're asking about a dominant trait, it's unless, they give some sort of stipulation that the dominant trait's only expressed when it's homozygous. But generally, it's going to be, if the dominant gene is there at all, it's probably going to be expressed. So good rule of thumb for Punnett squares. It's pretty much what I wrote down for genetics. Moving on into chemistry, yay! So we have the atom, which is the fundamental unit for chemistry. It's comprised of a nucleus, which holds the mass of the atom. So that's going to be made up of out of protons and neutrons. The nucleus is going to have a relatively like positive charge because that's where the protons are. Protons have positive charge, neutrons have neutral charge. And then the electron cloud is outside of the nucleus. That's basically where people, scientists think electrons are. We can't really pinpoint where electrons are at any given moment, but the electron cloud is basically like where we think the electrons are. And the electron cloud doesn't really have a mass, but it does have a negative charge because electrons have negative charge. So when looking at the periodic table, I don't know if this is going to be on it, but I felt like it was necessary to write this down. Remember that groups, or the columns of the periodic table, have the same number of valence electrons, and then the rows have the same like electron energy, or like the shells of the electrons. They have the same energy levels. That's what it's called. Or I just know as electron shells. Like they have the same amount of rings, basically. So basically the way that so when looking at like an element on the periodic table, you're going to see a few numbers. You're going to see the atomic number, which is the amount of protons. That doesn't change like at all. Electron, when the atom is in neutral state, is equal to the amount of protons. So for example, let's do helium, for example, which is 2. Helium has 2 protons because its atomic number is 2. And so that means when helium is in a neutral state, it also has 2 electrons. And then the number of, so you're also going to see the number underneath like the symbol, and that is the mass. So you're going to see that mainly to be like some sort of like number with like a decimal. The reason why it's a decimal is because that number is like the average isotope. So an isotope is basically when 2 atoms have the same amount of protons but different amounts of neutrons. So it's going to give them different masses, and the number that you see reflected on the periodic table is the average isotope based off of prevalence. So for example, if like carbon 12 is found naturally like 98% of the time, you're going to see a number very, very close to 12, but not quite 12 because there's also carbon 14 that exists. So like the number is going to be very close to 12, but like not 12. I mean, it's going to be very close to like what the most prevalent isotope is, but it's rarely going to be like a whole number. But for all intents and purposes, when you're calculating the amount of like neutrons in something, just like estimate, so like make it like 12 for carbon. I don't know like what the actual atomic mass is off the top of my head, like on the periodic table, but I know carbon is 12. So, so the way that you find the amount of neutrons in something is to look at the atomic mass and then subtract the atomic number from the atomic mass. What you're doing is subtracting the protons from the mass to find the amount of neutrons. Because like we said before, the nucleus holds the mass of an atom, and then that has protons and neutrons. So in order to find the amount of neutrons, you need to take out the protons, basically. So hopefully that made sense. So next we're going to go into bonding. So ionic bonding is when electrons are transferred. This is going to happen mainly between a metal and non-metal. So for example, sodium chloride, that's an ionic bond. So basically the only thing you really need to know about bonding is that when it's an anion, that means that it has a negative charge. So I remember an, like with an N, stands for negative. And then cation, cat has a T in it. So that's like, I think of a plus sign in my head. So that means it's going to be a positive ion. And then covalent bonding is a sharing of electrons. This is going to happen between 2 nonmetals. So for example, like water is hydrogen and oxygen. Those are both nonmetals. For covalent bonding, you might see a polar molecule. This means that it has a partial positive and negative charge on like either ends. So water is a great example. So it's kind of shaped like Mickey Mouse. So oxygen is here and you have 2 hydrogen atoms above it. So the side with the hydrogen atoms is going to be partially positive, and then the oxygen side is going to be slightly negative. Now we're going to go into states of matter, which is like super simple. So basically you have solid, liquid, gas. You also have plasma, but I doubt you're going to need to know that. So to go from solid to liquid is melting. So you're thinking of like water, for example. So like ice cube to water, you have to melt it. To go from water to like water vapor, you have to boil it. Or evaporate it. To go from gas, water, vapor to liquid, which is water, you have to condensate it. And then to go from liquid, water, to solid, ice, you'd have to freeze it. And then when you go from a solid straight to a gas and skip that liquid step, you're going to do something called deposition. And then the opposite is going from gas to solid. That's called sublimation, like when you're skipping that liquid step. So that's that. So now we're going to go into chemical and physical properties. So chemical properties are properties that can pretty much only be observed when you chemically alter a substance. So for example, if you burn something, you are, making that thing like combine with oxygen, but basically you're taking something out or putting something in something. So you're altering the chemical structure of something. So burning, rusting, flammability, testing something's corrosiveness or reactiveness, like those are all chemical properties. And then I think I got everything. Yeah. And then physical properties, these are things that can be observed without changing something chemically. So looking at something's color, shape, size, density, weight, mass, melting is still technically melting, boiling, like those are things that don't chemically change something. And the way that you can remember this is water is still water when it's ice, and water is still like water when it's liquid. So like it doesn't have the same physical form, but it's molecularly still the same thing. So then there are also intensive and extensive properties. Intensive properties are properties that I believe don't depend on the amount of something. Yes, so things like color, density, melting point, these are all things that are pretty much universal for something. It does not, the amount of something won't change those properties. And then extensive properties, so mass will change based on the amount of something. Volume will also change, and size. So things that, maybe like the amount of time something takes to melt, but not the melting point itself, those are extensive properties. Moving right along into chemical reactions. This is like my favorite part. So we have synthesis reactions, which are combining. So think A plus B yields AB. Decomposition reactions is taking something apart. So that's going to be like the opposite of synthesis. So you have AB yields A plus B. So you took apart A and B. Single replacement reactions is kind of self-explanatory. So that's going to look like AB plus C yields AC plus B. So in this example, B is being replaced by C, and that only happens to one element. And then double replacement is going to look like AB plus CD yields AC plus BD. So there's like a double swap there. And then combustion reactions is fuel, some sort of fuel, plus O2 yields water, I'm pretty sure, water, carbon dioxide, and some sort of energy, normally in the form of heat or light. Let me just double check. Yes. So that's another kind of reaction you have to watch out for. So if you see the product of something, or what the reaction yields to be water, CO2, and some sort of lighter heat, that should pretty much like flash a light bulb in your head and be like, oh, that's combustion. To go over, reactants are like what goes into the reaction, and then products are what come out of the reaction. Going over the other types of reactions, we have exothermic and endothermic. Exothermic reactions release energy, and then endothermic reactions absorb energy. So when you're looking at, if they show you like a graph and you see that the reactants have more energy than the products, you're going to know it's an exothermic reaction. Because if you think of it in terms of heat, you know, you have, think of like, you know, something getting colder. So we have like water to ice, right? You react, I don't know, this is like a bad example, but this is how I think of it in my head. So like water has like more heat to it than ice does. And so water is going to have to get rid of that energy in heat to make itself into ice. So that's like an exothermic reaction. I don't think the actual thing I described is like an exothermic reaction, but like the process is exothermic. And then same thing with endothermic. To go from like ice to like water vapor, the water vapor is going to need to absorb a lot of heat energy in order to get itself to that point. So hopefully that makes sense. So like if you see, if you know, like the kind of like graph that I'm talking about, like this kind of graph, it's shaped like this, then you know my pain. Because like looking at it, I was like, what is this? But once I was able to think of it that way, hopefully that helps. Oh, and then conditions that affect chemical reactions are going to be temperature, pressure, increasing the concentration, or I shouldn't say increasing, but concentration of the reactants or substrates, along with the presence of catalysts. So if you increase those things, you're going to like increase the like rate of the reaction. And then if you decrease those things, you're going to decrease the rate of the reaction. Moving on into acids and bases. So the thing that I learned in school was acids accept and bases donate, but you need to remember what they actually accept and donate, which are hydrogen ions. There you go. So also in the form of protons, because that's what a hydrogen ion is. Properties you need to remember about acids is that they have a sour taste. They turn litmus paper red. They produce salt precipitates when reacting with bases. They accept ions or hydrogen ions / protons, and and then bases, they're known as alkalines. That's important to know, and that's the opposite. So they're going to have like a bitter, soapy taste, bitter taste, soapy texture. They're going to turn litmus paper blue. They are going to donate their hydrogen ions or accept hydroxide ions. They produce hydroxide ions in solution, which is OH minus. And then we're going to go into strong acids and bases. So basically, highly reactive acids and bases are called strong because their atoms ionize in solution. So strong acids and bases, all of their atoms ionize. Weak acid bases, basically they're not as reactive, so only a few of their atoms ionize in solution. And then pH measures the amount of hydrogen ions in a solution. So the basically the lower the pH corresponds to how low those hydrogen ion levels are. So we know that acids accept those ions. That makes sense. So like a low pH is not going to have very many hydrogen ions because acids are accepting them. So if something's acidic, it has a low pH because acids are accepting those hydrogen ions. And then the reverse, if something has a lot of hydrogen ions, the solution is basic because bases donate those hydrogen ions. And then, so the pH scale goes from 0 to 14. Anything less than 7 is acidic, anything more than 7 is basic, and then 7 on the dot is neutral, basically water, basically. And then that was it for acids and bases. Now on to scientific reasoning, the last home stretch of the video. So basically what I wrote down, the scientific method is formulating a problem, forming a hypothesis, conducting an experiment, analyzing data, forming a conclusion. I don't know if I'm oversimplifying it, hopefully I'm not. And then things to know about scientific reasoning, what the groups and variables mean. So a control group is the normal state of a variable without being manipulated. And then the independent variable is the factor in the experiment that is being changed. The dependent variable is the factor in the experiment that is being measured. And then the constant are factors in the experiment that stay the same. So for like a classic do plants grow in the dark experiment, your control would be, in normal settings, plants, grow outside. So you would have, your plant outside in the sunlight all day. The independent variable would be light, right? Because you're changing the light situations. You might have a plant grow in the dark. You might have a plant grow in artificial light. You might have a plant grow in sunlight. And the thing that is actually being measured is plant growth. So that is your dependent variable. And then your constant would be factors in the experiment that stay the same. So maybe the plant type, the kind of soil that the plant is in, the pot that the plant is in, the amount of water that you give it, like all those things would be constants in experiments. So that was something that was like difficult for me to like remember, because the way that they explain independent and dependent variable, I was just like never able to absorb. But when I think about it this way, it's like easier. So that is it for part 1. Thank you so much for watching. So yeah, if you want to watch part 2, go right ahead. It should be up by Friday the 15th, because that's when I take my exam. So if you're watching this before Friday, October 15, 2021, I'm so sorry, you just have to wait like a few more days. But if you're watching this after, like into the future, November 2021 on, basically all the videos are up about the science section. So again, thank you so much for watching. If you want to see my face again, subscribe, and I'll see you next time.